Welcome, fellow goat enthusiasts, to Hoofnotes, where we make goat science make sense. I'm your host, and today we're embarking on a truly fascinating deep dive. We're going into a world you can't actually see, but one that, well, it profoundly impacts every single one of us in the goat community.
It really does.
Whether you're carefully hand-milking two beloved goats in your backyard, or, like me, here at Goat House Farm in sunny Florida, running a machine dairy for 22, this invisible force is at play, right there in your milkroom.
Absolutely.
We're talking about biofilms. Now, it's a term that might sound a bit academic, maybe a little like something from a science fiction novel.
It can sound that way.
But understanding it is absolutely crucial for the safety of your milk, the health of your goats, and just the smooth operation of your home dairy.
Couldn't agree more.
Our mission with this deep dive today is really to give you a genuine shortcut. We want you to be well-informed without feeling, bogged down by complex scientific jargon.
Yeah, cutting through the noise.
Exactly. We're going to peel back the layers, provide clear, actionable insights that will hopefully help you keep your milk pristine and your herd thriving. For me personally, spending a lot of time with my machine milker, always thinking about the best ways to care for my animals and ensure quality. The sheer science of bacteria, how they organize themselves, how they build these intricate little communities, it's truly captivating.
It is pretty amazing stuff.
It's like discovering a hidden micro-city right under your nose.
That's a great analogy, and we're thrilled to be your guides on this exploration. It's an often overlooked, yet immensely important aspect of dairy management.
So where are we pulling our info from today?
Well, for this deep dive, we've drawn insights from a really recent, impactful study. It was conducted on commercial dairy farms down in Chile.
Oh, interesting. Real-world data.
Exactly. And we're pairing that with comprehensive reviews of biofilm formation and control within the broader dairy industry. So it's a blend of, on-the-ground research and the overarching scientific understanding.
Perfect. So we get the practical and the science.
That's the goal, to give you a robust, fact-based understanding that you can immediately apply, really, no matter your scale. So let's unravel the mysteries of these bacterial fortresses.
Okay, so let's unpack this then. When we talk about biofilms, what are we really talking about? My initial thought, honestly, before I really started digging into this, was that it was just some kind of slippery, visible slime. The stuff you might see on a surface if you neglected it for too long.
That's a common starting point.
But is it just that? Or is there something far more complex, more organized, happening at a microscopic level? Something that makes these bacterial communities so incredibly special and, frankly, so persistent?
That's an important distinction to make. And yeah, it's far more intricate than just simple slime, though, like you said, a slimy appearance is certainly a hallmark.
Okay.
At its core, a biofilm is a highly structured, kind of self-organizing community of microorganisms. Now, while our primary focus today is on bacteria, these communities can also rope in yeasts and molds, too. So it becomes this diverse microbial ecosystem.
So not just one type of bug, necessarily?
Often not, no. But the defining characteristic, what really elevates it beyond just a random clump of microbes, is that these guys attach to a surface, they stick, and then, crucially, they produce their own self-made protective layer. This layer is called an exopolymeric matrix, or EPS for short.
EPS, okay, exopolymeric matrix. So that's the special sauce, so to speak. That's what makes it more than just microbes hanging out.
That's exactly it. It's the key difference. That EPS is what gives it structure and protection.
So it's almost like they're building their own miniature city walls, creating a fortified home for themselves.
That's a fantastic way to put it. Honestly, you can truly think of this EPS matrix as a fortress, or a city, or even like a self-constructed shield that these microbes build around themselves.
Wow.
It's slimy, yes, but it's also incredibly sticky and remarkably protective. And this matrix isn't a simple single substance either. It's a complex mixture, a cocktail really, of various biological materials.
Like what's it made of?
Primarily, it's made of these sticky polysaccharides, those are complex sugars, but it also incorporates proteins, lipids, which are fats, and even strands of their own extracellular DNA. They actually use their DNA as part of the building material.
Get DNA. It's wild.
Isn't it? This complex multi-component composition is precisely what gives the biofilm its structural integrity, its remarkable stickiness, and, well, its formidable resilience against external threats. Imagine trying to scrub off a sticky spider web that's also somehow reinforced with concrete and rebar that's kind of closer to the reality of a mature biofilm.
Okay, that paints a picture. Concrete spider webs. Definitely hard to clean.
Exactly. Now, what's particularly critical for us in the dairy world, whether you're a small homestead with just a couple of milking heads or a larger operation, is understanding why dairy processing facilities and equipment, including every single bit in our home milk rooms, provide such optimal conditions.
Right, wire stuff.
Well, they're just perfect environments for these microorganisms to not just survive, but to truly thrive and form these complex communities. There are very specific environmental factors at play.
Like,
First, you usually have consistent temperatures, often warm or room temperature, which are just ideal for bacterial growth. Then there's the constant presence of water and varying levels of humidity. Think about rinsing equipment, the moisture in the air.
always damp somewhere.
Precisely. But perhaps the most critical factor, the one that makes your milk room a veritable five-star hotel for microbes, is the abundance of organic material.
The milk itself.
Bingo. Specifically, milk residues, even tiny unseen traces of milk left on a surface like a microscopic film in a seam or a single drop left inside a hose, provide this rich, nutrient-dense food source. It basically encourages these bacteria to settle down, adhere, and just multiply like crazy.
And it sounds like they don't waste any time getting established either. It's not like these fortresses take weeks or months to build. You're saying they can pop up pretty quickly.
Oh, absolutely not. They don't hang around. And that's a really critical point when we talk about practical cleaning protocols later. Biofilm formation is not a slow, leisurely process. It's actually quite rapid.
How rapid are we talking?
Well, bacteria can begin to bind to surfaces within minutes of encountering them. Just think about how quickly a single drop of milk can dry and leave that little film on a surface.
Yeah, instantly almost.
Right. Once those initial bacteria attach, they immediately start secreting that protective EPS matrix we talked about. So the process of forming a fully mature, robust biofilm can develop within just a few days. And under really ideal conditions, plenty of food, moisture, the right temperature, it can even happen within a matter of hours.
Hours, wow.
Yeah. This rapid development is precisely why consistent, immediate, and effective cleaning protocols after every single milking are so vitally important. You're not just cleaning visible dirt, you're literally preventing the very early stages of a sophisticated bacterial colonization before it even gets going.
So these fortresses can pop up in a matter of hours or days. That's incredibly fast, and it definitely makes you think about how little time we actually have between milkings, right?
Exactly. The window is small.
If these communities are forming so quickly, what are the actual consequences for our milk and maybe more importantly for the health of our herd? Because that's where this really hits home for us goat owners.
Indeed. And this is where the theoretical becomes very practical and honestly a little concerning. The presence of biofilms on milking equipment represents a major and often quite persistent source of contamination for your milk.
How does that happen exactly?
Well, imagine it. You've got bacteria, maybe some yeasts and molds too, all comfortably nestled within this protective matrix on the internal surfaces of your lines, your buckets, your pulsator, maybe other machine parts.
Okay.
When milk flows through that equipment, especially with the turbulence and pressure changes that happen during milking.
Yeah, there's a lot going on in there.
Right. Pieces of that biofilm and all the bacteria encased within it can actually get dislodged, broken off. These then flow directly into and contaminate the collected milk. It's not just a few stray bacteria floating around. It's like a whole chunk of a bacterial city breaking off and falling right into your product.
That image of a whole bacterial city breaking off is pretty vivid and alarming. I think many listeners might be thinking, okay, but I always filter my milk really well, or maybe I pasteurize it later. Doesn't that take care of any problems from these biofilms? Is that a common misconception? Or are there still hidden dangers?
That's a fantastic and very common question. And it really speaks to critical nuance here. While filtering can certainly remove larger particles like sediment or hair, it's not going to remove microscopic bacteria. They just pass right through.
Makes sense.
And while pasteurization does kill a significant number of pathogens, that's its job, as we'll discuss a bit later, bacteria that originate from biofilms can actually become more resistant to heat.
Seriously?
Yeah.
More resistant?
Yes, it's a concerning finding. So while filtering and pasteurization are absolutely important steps for safety and quality, they might not entirely mitigate the risks that originate from these deeply entrenched biofilms on the equipment itself. The contamination source is still there.
Okay, so it's not a magic bullet. What are the direct consequences then in the milk itself?
Well, this direct contamination from biofilms leads to a number of undesirable consequences. Most notably, it results in increased overall bacteria counts in your milk. Now, while some bacteria might be harmless spoilage organisms, high counts are always a clear indicator of a quality issue.
Right. Higher counts aren't good.
And they can suggest the presence of those spoilage organisms, or worse, actual pathogens. This higher bacterial load can lead pretty quickly to undesirable changes in the milk itself. Think about the rapid development of off flavors, anything from sour to bitter, or even that kind of barny taste that nobody wants.
Definitely not in my fresh goat milk.
Exactly. Nobody wants that in their delicious, hard-earned goat milk. And crucially, it also significantly shortens the shelf life of your raw milk. It just won't stay fresh and palatable for as long as it should, meaning you have to use it or process it much sooner than you'd like.
That's a big deal for home use, too. You want it to last.
Absolutely. But the implications, unfortunately, extend far beyond just milk quality. Biofilms can become really dangerous on-farm reservoirs for pathogenic bacteria,
Pathogenic meaning.
meaning bacteria that can cause serious disease, both in animals and, importantly, in humans who might consume or handle the milk, or even just interact with the farm environment.
Okay, this is getting serious.
It is, and this is a critical risk, especially for udder health in your does. Studies have specifically highlighted that biofilms found, for example, in the rubber liners on milking machine claws, those parts that are in direct, intimate contact with the teats.
Yeah, right on the business end.
Exactly. Those can be a persistent source of bacteria, like Staphylococcus aureus. Now, when these contaminated liners come into close contact with the teat during milking, it poses a significant risk of intramammary infections, which we commonly know as mastitis.
Oh, mastitis. Every goat owner dreads that.
Exactly. We all know how devastating and difficult mastitis can be for our goats. It causes pain, definitely reduces milk production, impacts quality, and sometimes can even lead to the permanent loss of a quarter. So the link between equipment biofilm and mastitis is a really critical one.
That's a huge concern for anyone milking, especially with a machine where those rubber liners are in constant repeated contact. And I remember reading in the materials about a specific, often overlooked hotspot in these systems, something that seems to fly under the radar. What was that about?
Yes. You're probably referring to the really illuminating findings from that Chilean dairy farm study we mentioned earlier.
Yeah.
It spotlighted a significant and often seriously neglected area for biofilm presence. The hoses used to divert waste milk, colostrum, and transition milk.
Okay, so the milk that doesn't go in the main tank, why is that such a hotspot?
Well, this is the milk from animals that might be undergoing antibiotic treatments, or maybe they have mastitis, or even just fresh that haven't been cleared for the main bulk tank yet. The rationale behind neglecting the cleaning of these specific hoses is often, well, this milk isn't for sale, it's just going to the calves, so it doesn't need the same super rigorous cleaning.
I can see how people might think that, but
But it's a really dangerous oversight. Because this diverted milk isn't destined for commercial sale, the cleaning protocols for these specific hoses are frequently just overlooked, or they're simply not as thorough as those for the main milking lines. And this neglect creates absolutely ideal, undisturbed conditions for biofilm formation and serious growth.
makes sense. Less cleaning equals more biofilm. What did they find in those hoses?
In that Chilean study, the researchers actually isolated and detailed several specific pathogenic bacteria from these neglected hoses. They found Staphylococcus aureus, which we just talked about with mastitis. They also found Pseudomonas aeruginosa, Klebsiella pneumonia, and Klebsiella oxytoca.
Okay, those names sound vaguely familiar and vaguely ominous.
They should. These are not benign organisms. They are serious opportunistic pathogens. They're capable of causing significant illness, especially in vulnerable animals.
And the most concerning part is that this waste milk, or colostrum, it often isn't actually wasted, is it? It's frequently fed to calves, or in our case, sometimes goat kids.
That's exactly right.
So we're essentially feeding our most vulnerable animal, the babies, through highly contaminated equipment. That just feels fundamentally wrong when you think about it that way.
It is fundamentally wrong, and it really highlights a critical potential breakdown in biosecurity and animal welfare practices on some farms. Feeding calves or kids this waste milk or colostrum collected through contaminated equipment directly exposes these young, often immune-compromised animals to a potentially high load of these harmful pathogens.
What kind of illnesses are we talking about from these specific bacteria?
Okay, let's break it down. To give you some concrete examples, Staphylococcus aureus. We mentioned mastitis, but in young animals, it can actually lead to intramammary infections developing in heifers even before they start milking, particularly if there's cross-suckling among calves or kids sharing pens. It can also cause other skin or localized infections. Klebsiella species like K. pneumonia and K. oxytoca, these are particularly concerning for young stock. They are frequently implicated in severe respiratory infections like pneumonia, which we know is a major cause of calf and kid morbidity and mortality.
Pneumonia, yeah, that's a big one.
And they can even cause more severe systemic conditions like meningoencephalitis. That's an inflammation of the brain and its surrounding membranes in bovines, and potentially similar neurological issues in goats. Really nasty stuff. Pseudomonas species. These are kind of versatile troublemakers. They're associated with a range of infections in calves and kids, including respiratory infections, urinary tract infections, and ear infections. And adding insult to injury, they're often particularly difficult to treat due to inherent resistance to many common antibiotics.
Wow, so a whole range of potential problems just from using dirty dump lines.
Exactly. And the real-world impact of this neglect was powerfully demonstrated in that Chilean study. They noted that Farm A, which had a significant and abundant biofilm presence detected in its diverted milk hoses, also frequently reported alarmingly high rates of calf morbidity and mortality.
What were the calves dying from?
The primary causes being reported were diarrhea and pneumonia, the very things these bacteria can cause. Now, while other management factors might contribute, the study strongly suggests a direct and pretty undeniable link between the contaminated milk being fed to those vulnerable calves and the high rates of illness.
That's truly sobering. It really underscores that what we might consider waste milk needs just as much care and handling as the milk we drink.
Absolutely. It can have very real, very negative consequences if not handled with the same diligence.
And this isn't just an on-farm animal health issue then, is it? What about public health implications? Even for us, small home dairies who aren't selling milk commercially. Maybe we're just consuming it ourselves or sharing with friends and family.
Right.
Does it still matter?
Yes, it absolutely still matters. Even for home dairies where the milk is consumed by family or close friends, there are significant public health implications that really shouldn't be overlooked. These pathogens from biofilms, they aren't necessarily contained just to the equipment or the milk itself.
How so? How do they spread?
Well, they can spread throughout the farm environment through various means, creating a much broader contamination risk. For instance, fecal shedding. Calves or kids that have been fed contaminated milk can actually shed these pathogens in their feces. This contaminates bedding, feed troughs, water buckets, the general pen environment.
Okay, so it gets everywhere.
Pretty much. Then there's direct contact. Dairy personnel, including us home dairy owners, who handle contaminated equipment, maybe touch a dirty hose, then touch a gate latch, or your boots track it around, can inadvertently transfer these bacteria to other animals, to other clean equipment, or even potentially into the home environment on hands, clothing, or footwear.
Right, basic biosecurity.
Exactly. And don't forget environmental vectors. Even seemingly innocuous things like insects, particularly flies, can act as mechanical vectors. They land on a contaminated surface, pick up bacteria, and then fly off to land on clean surfaces, feed, or directly onto animals.
Ugh, flies. The bane of my existence.
Tell me about it. So, while you might not be selling commercially, you're still potentially creating a reservoir for pathogens on your property that could impact your family or anyone else who interacts with your farm.
That's a really important perspective. And there was another risk mentioned, something even more concerning maybe, about antibiotics.
Yes, and this is a really serious, often overlooked, and frankly deeply concerning risk related to antibiotic resistance. Think about it. When milk from does undergoing antibiotic treatments is diverted through equipment that already harbors biofilms.
Which we just established happens in those neglected hoses.
Precisely. The bacteria living within those biofilms are then continuously exposed to antibiotic residues present in that milk. Now, this isn't usually enough to kill them outright, especially inside the protective biofilm, but it's enough to create this constant sublethal exposure.
What does that do?
It creates an absolutely ideal selective environment for bacteria to adapt and develop resistance. It's like a low-dose training camp for bacteria, basically teaching them how to survive our most powerful medications.
Well, that's not good.
Not good at all. This continuous pressure selects for and actively drives the development and proliferation of antimicrobial resistant bacteria within the biofilms. Once these resistant strains develop, they don't just stay put. They can then potentially spread to other animals on the farm, into the wider environment through manure application perhaps, and even indirectly to humans.
Contributing to the bigger superbug problem.
Exactly. It contributes significantly to the broader and increasingly urgent global challenge of antimicrobial resistance, which affects all of us by making antibiotics less effective when we truly need them. The Chilean study also specifically noted that waste milk containing these antibiotic residues can directly cause issues like diarrhea and unhealthy changes in the gut microbiome of the calves drinking it, further highlighting why we need to limit their exposure.
Okay, so this is heavy stuff. Biofilms contaminating milk, making our goats sick, potentially spreading disease to people, and contributing to antibiotic resistance. If they're causing so many problems, why aren't they easy to just scrub away or kill with a simple disinfectant? What makes them so incredibly stubborn and hard to eliminate once they've really established themselves? It honestly feels like we're fighting an uphill battle sometimes.
That's because, in many ways, you truly are dealing with a formidable opponent. It's not just a feeling, it's a scientific reality. What makes biofilms so remarkably resistant to our cleaning and sanitizing efforts really comes down to several sophisticated, kind of multi-layered defense mechanisms they employ.
Okay, hit me with the defenses. What's strategy number one?
First, there's that physical barrier we keep mentioning, the exopolymeric matrix, the EPS. Think of it again as that thick, almost impenetrable shield, like biological concrete.
Like the fortress wall.
Exactly. This EPS matrix acts as a really formidable physical barrier. It actively prevents or significantly slows down the movement of detergents, disinfectants, and even antibiotics into the inner layers of the biofilm. It literally acts like a protective suit, shielding the bacteria nestled deep within.
So the cleaners can't even get to them.
Often, they can't reach the inner layers at effective concentrations. They simply can't penetrate that sticky, gooey, yet incredibly tough barrier effectively enough to kill everything inside.
Wow. So it's not just that the bacteria themselves are tougher, it's that the cleaning agents can't even reach them properly. That's quite a strategy. They're like microscopic ninjas in an armored suit.
That's a great way to think about it. Exactly. And it gets even more complex than that. Within this protective fortress, the bacteria aren't just isolated individuals passively waiting for their fate. No, they're highly organized. They actually communicate with each other.
Bacteria communicate. How?
They use a really sophisticated cell-to-cell signaling system. It's called quorum sensing, or QS for short.
Quorum sensing. Like needing a certain number to vote.
Kind of. Imagine these bacteria like tiny spies, constantly releasing and detecting specific chemical messages, they're called autoinducers, into their immediate environment. When a certain concentration of these chemical signals is reached, that's their quorum, meaning a sufficient number of bacteria are present. And signaling it essentially tells the whole community, okay, gang, there are enough of us here. Time to initiate our collective defenses, coordinate activities like producing enzymes to break down stuff, and maybe activate some resistance genes.
That's incredible. They talk to each other to coordinate their defenses.
Absolutely. This coordinated communication allows the multi-species community within the biofilm to act as a unified, highly protected, and incredibly resilient entity. This collective action makes them much, much harder to remove or destroy than individual free-floating bacteria just drifting around. They act as one, and that makes them stronger together.
Okay, so physical barrier, check. Communication network, check. What else have they got?
Beyond that, the bacteria living within biofilms undergo significant physiological adaptations. They basically adopt a distinct phenotype, meaning their behavior and characteristics change dramatically compared to their free-floating counterparts, the ones we call planktonic.
How do they change?
For instance, they can shift into a sort of slow growth state. This often happens because nutrients might become limited deep inside the dense biofilm, or maybe their own waste products start to accumulate. Now, when bacteria are growing slowly, they're inherently less susceptible to many antimicrobials.
Why is that?
Because many cleaning agents and antibiotics are specifically designed to target actively growing, metabolically active cells. They might interfere with cell wall synthesis or protein production, for example, processes that are significantly slowed down or minimized in these slow-growing biofilm cells.
Clever. So they just slow down to ride out the storm.
Pretty much. And furthermore, some bacteria within biofilms can even differentiate into these unique, highly resistant cells called persister cells.
Persister cells.
Yeah. These are incredibly tough. They enter a dormant, non-dividing state, almost resembling bacterial spores in their resilience. They are highly tolerant to extreme conditions, including many antibiotics and disinfectants. They essentially serve as a sort of microbial seed bank within the biofilm.
As a seed bank.
Exactly. Yeah. They can survive even a harsh cleaning attempt that kills most of the other cells, and then, once conditions become favorable again, maybe when the cleaner is rinsed away, these persister cells can wake up and reinitiate the biofilm growth all over again. This makes completely eliminating them extremely challenging.
Wow. Okay, this sounds like a masterclass in survival.
Hmm.
When you put all these factors together, the physical barrier, the communication network, the physiological changes, these persister cells, how much more resistant are these biofilm bacteria compared to the ones that are just floating around freely in milk or water? It really feels like we're outmatched.
The increase in resistance is truly dramatic, and it's precisely why we need to be so incredibly diligent with cleaning. Bacteria living within mature biofilms can be anywhere from 10-fold to an astonishing 1,000-fold more resistant to common antibiotics and biocides compared to their free-floating or planktonic counterparts.
10 to 1,000 times. That's huge.
It is absolutely huge. This incredible resilience is precisely why traditional, sort of superficial cleaning methods often fail to completely eradicate them. You might kill 99% of the free bacteria, maybe even some on the surface of the biofilm, but those protected survivors deep inside and those persister cells, they remain to regrow.
Okay, that number really drives it home. But wait, there was something else mentioned, something about temperature. That sounded surprising.
Yes, and this is also incredibly important, especially for dairy operations. There are findings related to temperature that are quite counterintuitive. Firstly, biofilm formation can actually occur, and sometimes even increase, when bacteria are held in cold storage conditions, like at 5 degrees C, which is around 40 degrees Apatol, a common temperature for raw milk storage.
So putting milk in the fridge doesn't stop them from forming biofilms. It might even encourage it sometimes.
In some cases, yes. The cold can stress certain bacteria in a way that triggers biofilm formation as a protective response. But even more critically, bacteria that form these biofilms in cold temperatures can actually become more tolerant to subsequent heat treatments, like pasteurization.
Wait, hang on. They become more resistant to heat if they form the biofilm in the cold. That makes absolutely no sense. How does that work? So even if someone later pasteurizes their milk, these biofilm-derived bacteria could potentially survive and still cause issues. That feels like a double whammy.
It is completely counterintuitive, isn't it? But it's a significant scientific finding with very real-world implications for dairy product safety. The mechanism behind this increased heat tolerance is fascinating, actually.
Okay, explain this one.
These biofilm-dwelling cells, particularly when they form their communities in cold conditions, show an increased expression of what scientists call heat shock-related genes. Think of these genes as the blueprints for tiny, robust cellular repair crews inside the bacteria.
Heat-shock genes. Okay.
These genes produce specialized proteins. They act like molecular chaperones, essentially helping to fold and stabilize other crucial proteins within the bacterial cell, preventing them from denaturing or breaking down when exposed to high heat. It's like giving the bacteria its own internal protective shield or repair kit.
So they're pre-programmed to handle the heat better if they grew up in the cold biofilm.
That's a good way to put it. It allows them to survive thermal processes like pasteurization that would typically eliminate their free-floating counterparts that haven't built up these defenses. This phenomenon helps explain why certain spoilage organisms, and sometimes even some pathogens, can persist even in commercially processed dairy products. It means a problem that starts way back in your milking equipment biofilm can potentially follow through all the way to your finished product, even after heating.
That is genuinely mind-blowing and worrying.
It is. And finally, just to reiterate and really emphasize that point we touched on earlier about antibiotics, that continuous exposure of biofilm-dwelling bacteria to antibiotic residues, especially in that diverted milk scenario.
Right, the training camp.
Exactly. It provides that ideal selective environment. This constant presence of antibiotics really amplifies the risk of selecting for and promoting the proliferation of antimicrobial resistant strains within those protected biofilms. It is truly a prime breeding ground for superbugs.
Making it not just a farm problem, but a public health problem.
Absolutely. It's a critical factor not only for the immediate health of your herd, but also for the much broader public health concern of antibiotic resistance, as these resistant genes can potentially be shared with other bacteria and transferred through the environment.
Okay, deep breath. So we know biofilms are tough, truly tenacious adversaries, but we are absolutely capable of tackling them. The goal here isn't just clean enough, it's what I like to call kitchen clean.
I like that standard.
Think about how you want your dinner plates and forks to be before you eat off them, right? That level of spotless, squeaky clean perfection. So what's our step-by-step game plan? For milking equipment, whether it's a simple hand milker, a small bucket milker, or a more complex machine setup like mine at Goat House Farm, how do we actually achieve that truly kitchen clean result consistently? every single time.
That kitchen clean philosophy is the perfect mindset to approach this. It really is. It means that after every single milking, your equipment should be as visually clean and, just as importantly, as dry as the dishes in your kitchen drawer, free from any visible residue, any dull film, or any lingering moisture.
And not just clean looking, but actually clean.
Precisely. And it's important to stress that kitchen clean doesn't mean you have to resort to overdoing it with harsh, excessive chemicals. Sometimes people think stronger is always better.
Right, more soap, more bleach.
Yeah, but using super strong concentrations or simply too much cleaner can actually degrade your equipment over time, especially rubber and plastic parts, shortening its lifespan. And it could potentially leave unhealthy residues in your milk, which nobody wants. The real goal is to find the right balance for effective, consistent sanitation, combining good mechanical action scrubbing with the appropriate chemical agents at the right concentrations and temperatures.
Okay, makes sense. Balance is key. So walk us through it. What are the essential steps for the daily cleaning routine, and why do we do each one?
All right, let's break it down. The first critical step, and honestly one that is often underestimated or done incorrectly, is step 1, the critical lukewarm water flush. This is your absolute first move immediately after you finish milking. Don't let things sit.
Okay, immediate lukewarm flush.
Yeah.
Why lukewarm?
Purpose. The primary purpose here is simple, but absolutely vital. To rinse away all that visible milk and the gross organic debris before it has any chance to dry onto the surfaces. You want to get rid of as much of the bulk milk residue as possible right away. Temperature is key, and the emphasis is on lukewarm water. This isn't just a casual suggestion. It's crucial because of the chemistry of milk. If the water is too hot, generally above about 120 degrees Fair or 49 degrees C, the delicate milk proteins, particularly casein, can actually coagulate. They basically cook onto the surfaces of your equipment.
Ah, yes. The scrambled egg effect.
Exactly. Think of trying to clean scrambled eggs off a pan after they've baked on it becomes incredibly difficult, almost impossible sometimes. Conversely, if the water is too cold, the milk fats might solidify and stick to the equipment, forming this greasy, hard-to-remove film.
Okay, so lukewarm is the sweet spot. Goldilocks temperature.
Precisely. Lukewarm water allows both the proteins and the fats to remain suspended and flow freely, so they rinse away easily. A really common mistake is people blasting it with scalding hot water right away, thinking they're sanitizing early. But instead, they often just set themselves up for a much harder cleaning challenge later by cooking that protein on.
That makes so much sense. I've definitely learned that lesson the hard way, trying to use hot water too soon. It was exactly like trying to clean scrambled eggs off the inside of my milk bucket. A very memorable, if messy, lesson in protein coagulation. Okay, so lukewarm rinse first. What's next after that crucial initial step?
All right, after that essential lukewarm flush, where you've hopefully removed the bulk of the milk solids, you move on to step 2, the hot alkaline wash.
Hot and alkaline. Okay, why?
Purpose. The purpose of this wash is twofold. First, it's designed to remove the remaining fats, proteins, and also minerals that might still be clinging to the equipment surfaces, often forming a thin, almost invisible film. Second, and importantly for our topic today, this step really begins the process of attacking and breaking down any nascent or newly forming biofilms.
So this is where the real cleaning happens. Why alkaline?
Most dedicated dairy detergents and soaps you'll find are alkaline, meaning they have a high pH. They are specifically designed for this task because alkaline solutions are highly effective at dissolving both fats, saponification, and proteins, hydrolysis. Look for cleaners specifically formulated for dairy use. They often contain other helpful agents like chelating agents to help remove mineral deposits as well.
Okay, dairy-specific alkaline cleaner. Does how we apply it matter?
Yes, the method of application varies a bit depending on your setup. For hand milkers and bucket milkers, this absolutely means complete disassembly of all parts that touched milk. Every single component, the inflations, the hoses, the lid, any pulsator attachments, the bucket itself, the strainer, the filter holder, everything should be taken apart.
Everything comes apart every time.
Every single time. These individual pieces then need to be manually scrubbed with hot soapy water. You absolutely need a dedicated set of brushes just for your milkroom. Don't use your kitchen sponge. And include brushes specifically designed for cleaning inside hoses and those inflation tubes. Make sure you physically reach every internal surface. Even if you use a little pump to circulate cleaning solution through the hoses for a bucket milker setup, that manual scrubbing of each piece is still non-negotiable for thoroughness. You have to physically dislodge any adhered material. For pipeline milking systems, like the one you use at Goat House Farm, these more automated systems rely heavily on the power of turbulent flow of the hot cleaning solution. It's not just about filling the lines with hot water, it's about the vigorous, high-velocity scrubbing action of the moving water and detergent mixture against all the internal surfaces.
Right, the force of the water does the scrubbing.
Exactly. Achieving proper turbulence is critical for physically scouring away residues and preventing biofilm attachment points from forming. And there's a critical temperature check here. The cleaning solution must be maintained at least 120 degrees F, 49 degrees C, preferably hotter at the start, like 160, 170 degrees F, but it needs to be at least 120 degrees F at the outlet of the system, meaning at the very end of the wash cycle.
Why is the outlet temperature so important?
Because that ensures it's hot enough to be effective throughout the entire cleaning cycle, even after losing some heat as it travels through the pipes. And crucially, maintaining that heat prevents dissolved milk solids from cooling down and readhering to the pipes as the water cools. If your wash solution cools too quickly, you risk just moving the dirt around and redepositing dissolved milk solids further down the line.
Got it. Hot alkaline wash, manual scrubbing or turbulent flow, keep it hot. What's step 3?
Next, you have step 3. A thorough water rinse.
Just rinse again. Why?
Purpose. The purpose here is straightforward, but absolutely essential. To rinse away all of the alkaline cleaning chemicals and any remaining loosened debris completely. You absolutely do not want any detergent residues left on your equipment.
What happens if you leave detergent?
Well, they can affect the taste of your milk, they can potentially interfere with subsequent sanitization steps like an acid rinse, or they could even create a slight film that ironically encourages bacterial regrowth. So this rinse needs to be as thorough as the initial flush, using plenty of water, ensuring everything is flushed clear until the water runs clean.
Okay, rinse away all the soap. Makes sense. Then step 4.
Following that thorough rinse comes step 4, the warm acid rinse.
Acid. Why acid now?
Purpose. This step is often overlooked, especially in smaller home dairies, but it is incredibly important for long-term bacterial control and also for preventing mineral buildup. Most bacteria, including those pesky ones that form biofilms, really thrive in neutral pH environments. That's where they feel most comfortable and multiply most rapidly.
So the acid makes it uncomfortable for them.
Exactly. An acid rinse, ideally one that results in a surface pH of about 3-4, serves two main purposes. First, it lowers the pH of the equipment surfaces, creating an environment that is distinctly unfavorable for bacterial growth between milkings. It effectively inhibits their proliferation until the next time you use the equipment. Second, acid rinses are excellent at dissolving and preventing the buildup of milkstone.
Milkstone. What's that?
Milkstone is those stubborn mineral deposits, mostly calcium and magnesium phosphates from the milk itself, combined with some protein and fat that can build up over time, especially if you have hard water. These deposits create rough surfaces on your equipment, and those rough surfaces are perfect little havens, ideal anchoring points for biofilms to get started and hide from cleaning. Using an acid wash regularly keeps your stainless steel and other surfaces smoother and much less hospitable to bacterial attachment.
Okay. So acid rinse inhibits bacteria and prevents mineral buildup. Good combo. The temperature for this one. Warm.
Yes, typically warm, not hot. Follow the manufacturer's instructions for the specific acid cleaner you're using regarding temperature and concentration. You don't need it scalding hot like the alkaline wash.
Okay. Alkaline wash, rinse, acid rinse. Are we done yet?
Almost. One more crucial step for the daily routine. Step 5. Complete drying.
Drying. You mentioned this earlier. Why is it so critical?
Purpose. This step is absolutely fundamentally crucial because moisture is the ultimate breeding ground for bacteria. Any residual water left on or inside your equipment, even a tiny droplet lingering in a crevice or hose bend, creates a perfectly hospitable environment for microbial growth. It can effectively negate all your previous diligent cleaning efforts. Bacteria simply need moisture to grow and multiply. Take away the water and you significantly slow them down or stop them between milkings.
Makes sense. No water, no life, basically. How do we make sure it's completely dry?
Methods. For hand milkers and bucket milkers, this means not just giving them a quick wipe down, but truly allowing them to air dry completely. The best practice is to invert milk buckets, hoses, and inflations. Hang them on dedicated racks or pegs, not just laid on a counter, so air can circulate freely both inside and out.
So hanging them up is better than just setting them upside down.
Yes, ideally, because it allows better airflow all around and inside. Never stack parts or store them flat while they're still wet. Even a small puddle of water collecting in a hose band or the bottom of an inverted bucket becomes a perfect stagnant little petri dish for bacteria to thrive in the hours between milkings. Consider using an air dryer attachment if you have a machine system, or simply ensure your milkroom or drying area has excellent ventilation and low humidity. The ultimate goal is to eliminate all residual moisture before the next use. This step, honestly, is where many people fall short, unknowingly sabotaging all their excellent cleaning work by leaving a moist haven for microbes.
Yeah, I can definitely attest to the importance of drying. Nothing feels worse than going to milk and finding that little puddle of water in a hose or bucket from the day before. It's just a gut punch to all your hard work.
Exactly. And for bulk tanks and their valve parts, these should also be completely disassembled, especially the valve, and allowed to dry thoroughly between uses, perhaps even wiped dry with a clean, dedicated cloth, ensuring no moisture remains trapped in threads or seals. For the more complex pipeline systems, good sanitary design should inherently minimize low points where moisture can accumulate, making gravity drying more efficient. But if you do have any known low points in your system, you need to make sure they are specifically drained and ideally blown out with clean air to ensure they dry completely.
Okay, dry, dry, dry. Got it. Now, you mentioned dead zones earlier as biofilm hotspots. Where specifically do we need to be extra vigilant about those sneaky spots?
Ah, yes, those dead zones. They are precisely where biofilms love to hide and become incredibly persistent because they're hard to clean effectively. These are basically any areas where the cleaning solutions don't flow with full force or turbulence, or where moisture tends to accumulate and linger, or simply where surfaces are not perfectly smooth. Think of them as the dark, quiet corners or the rough patches of your milking system where bacteria can set up shop undisturbed.
Give me some examples. Where should I look?
Specific examples to pay close attention to include tight bends and awkward joints in hoses and pipes where flow might slow down, rubber gaskets and O-rings. These are notorious. They often have tiny imperfections. They can compress over time, creating crevices, or they degrade, becoming porous. Valves and spigots, especially the internal mechanisms, which are often complex with multiple small parts, threads, and seals that are hard to reach effectively with cleaning solutions or brushes. Valves and spigots, cracks and scratches on your equipment surfaces, even microscopic scratches on stainless steel or plastic provide ideal anchoring points for bacteria to latch onto. Valves and spigots, any low points in your system, as we just discussed, where water can pool instead of draining completely. Valves and spigots. Also, sometimes unpolished welds inside stainless steel equipment can be rougher than the surrounding surface and harbor bacteria.
Okay, lots of potential hiding places.
Definitely. Biofilms will happily colonize these protected niches because they offer shelter from the full force of the cleaning solution's flow and often provide a steady supply of trapped moisture and residual nutrients. It's absolutely essential that these areas receive special attention during cleaning.
Like taking things apart.
Exactly. For example, that valve on your bulk tank. It should be completely disassembled, every single part, scrubbed, cleaned, and rinsed every single time the tank is emptied. Don't just rinse around the outside of it, take it fully apart and ensure every component is spotless and dries properly. Any area that milk touches and isn't perfectly smooth and directly exposed to the full force of the cleaning solution's flow is a potential hotspot that demands your vigilant attention and often manual intervention.
That makes sense. And the equipment itself matters too, right? The material.
Oh, absolutely. The material and the design of your equipment significantly impact how easily and effectively it can be cleaned. High-quality, smooth materials like food-grade stainless steel, especially 304, 316 grade, are always preferred because they offer fewer microscopic imperfections for bacteria to cling to and are resistant to corrosion from cleaners.
What about plastics or rubber?
Plastics can be okay if they're food-grade and kept smooth, but they scratch more easily than stainless steel, creating hiding spots. And rubber components like inflations and hoses wear out. As they age, they can develop cracks and become porous, making them much harder to clean and sanitize effectively. They need regular inspection and replacement. Conversely, using scratched, poor-quality plastics or old, worn-out rubber components can become an absolute nightmare for biofilm control.
So invest in good quality materials and replace worn parts.
Exactly. And ideally, equipment should be designed with cleanability in mind, avoiding those dead ends, sharp internal angles, and intricate crevices where dirt and bacteria can accumulate and biofilms can form, making them much harder to clean effectively. Good sanitary design really works hand in hand with good cleaning protocols. It's an upfront investment that definitely pays off in better milk quality and improved herd health down the line.
Okay, so we've totally nailed the daily cleaning routine now. The flush, the alkaline wash, the rinse, the acid rinse, and the crucial drying. Making sure everything is kitchen clean and bone dry. That's our solid foundation. But knowing how incredibly resilient these biofilms are and how sneaky they can be in those dead zones. What are some advanced tactics, some long-term strategies we can use to ensure our milkroom stays as clean and as safe as possible? How do we really go that extra mile? What are the pro tips for biofilm management?
That's a great forward-thinking approach, asking about the pro tips beyond the daily grind. Because you're right, even with excellent daily cleaning, biofilms can sometimes still gain a foothold over time. So beyond that daily routine, there are indeed some advanced strategies we can employ to manage them effectively and really prevent them from becoming established long term.
Okay, I'm ready. What's the first pro tip?
One key tactic is rotating your cleaners. We sometimes call this the shock approach.
Shocking the system. How does that work?
Well, biofilms, like any resilient adversary, can actually adapt and potentially develop some level of resistance or tolerance to specific cleaners if you use the exact same chemicals day in and day out indefinitely. This happens due to the inherent adaptability of microbial communities that are survivors.
So they get used to our routine.
They can to some extent. So to prevent this resistance from building up, we recommend periodically shocking your system. Ideally, maybe about once a month or so, you should consciously rotate the types of alkaline and acid cleaners you use.
Give me an example.
Okay, for instance, if you normally use a chlorinated alkaline cleaner, you might switch to a non-chlorinated alkaline cleaner for a month. Or maybe you alternate between an acid cleaner based on phosphoric acid and one based on nitric acid, if appropriate for your equipment. This rotation introduces different chemical stressors and modes of action to any lurking biofilm community, making it much harder for them to develop broad resistance mechanisms and generally keeping them off balance.
That's interesting. It's like changing up your workout routine so your muscles don't get too comfortable and plateau. You're doing the same thing to your bacterial adversaries. Keep them guessing.
Exactly. That's a perfect analogy. You're preventing them from becoming complacent or adapting too well to one specific challenge.
Okay, rotate cleaners monthly. Got it. Any cleaners we should avoid?
Yes, actually. Speaking of cleaners, an important note of caution. We strongly advise against using quaternary ammonium compounds, often called QACs or quats, for cleaning milking equipment surfaces that come into direct contact with milk.
QACs. Why not? They seem common in other cleaning products.
They are powerful sanitizers, yes, and you see them in many household and some farm disinfectants. However, they are notoriously difficult to rinse away completely from surfaces, especially porous ones or complex equipment. They tend to leave persistent residues.
And residues are bad because...
These QAC residues can cause unpleasant off flavors in your milk. They can also inhibit the starter cultures you might need if you're making cheese or yogurt. And there are some ongoing discussions and potential concerns about the health implications of ingesting QAC residues regularly. So for milking equipment, it's best to stick with effective, easily rinsed, dairy-approved alkaline and acid detergents and appropriate sanitizers when needed. Avoid the quats for milk contact surfaces.
Okay, noted. Avoid QACs on milk lines. What's the next advanced strategy?
Next, let's talk about strategic sanitizing. Now, while the daily cleaning routine we outlined is the absolute bedrock, adding a sanitizing step just before milking can be a valuable extra layer of security. While it's sometimes listed as optional for certain systems or if cleaning is impeccable, it's highly recommended for many situations, especially for small dairies aiming for the lowest possible risk of contamination and certainly for those aiming for top-tier raw milk quality.
So cleaning removes the dirt and food, sanitizing kills the leftover bugs.
Essentially, yes. Purpose. The main purpose of a sanitizer applied to already clean surfaces is to inactivate or kill any bacteria that might have survived the cleaning process or, more likely, grown in the system between milkings despite your thorough cleaning and drying efforts. It's basically your last line of defense right before the next milking begins. Sanitizers achieve this by rapidly killing or inactivating microorganisms on contact on those clean surfaces.
When should we definitely consider sanitizing?
When to consider. Sanitization is especially important for pipeline systems that are only used once per day. That longer downtime simply allows more time for any surviving bacteria to potentially multiply. It's also highly beneficial during periods of a high heat and humidity like you experience much of the year in Florida.
Oh, yeah. Tell me about it.
Because those conditions significantly promote bacterial growth and can really hinder the proper complete drying of equipment, making sanitizing more critical. Additionally, it's worth noting again that some states may have specific regulations that legally require pre-milking sanitization for licensed raw milk production. So always check your local state and county regulations.
Good point. Check local rules. How do we use sanitizers correctly? I worry about residues.
Proper use is critical. That's a valid concern, and it's the most important rule with sanitizers. Use no more than the recommended amount specified on the product label for a no-rinse application on food contact surfaces. Using too much absolutely can lead to harmful or taste-altering residues ending up in your milk.
So follow the label exactly. What about using something common like bleach?
If you're using standard household bleach, which is sodium hypochlorite, as a no-rinse sanitizer, the common recommendation, often cited based on federal guidelines for food contact surfaces, is typically no more than about 1 tablespoon per gallon of water for standard 5.25% sodium hypochlorite bleach. That yields about 100-200 parts per million ppm available chlorine. However, bleach concentrations vary significantly between brands and even product lines now.
Oh, right. Some are concentrated now.
Exactly. Some common disinfecting bleaches are now 7.5% or even 8.25% sodium hypochlorite. Using the same 1 tablespoon per gallon of that stronger bleach would result in a much higher, potentially unsafe concentration. So, you must check the label of your specific bleach product. Better yet, the best practice is to use chlorine test strips.
Test strips, like for a pool.
Similar idea, yes, but designed for sanitizing solutions. They're inexpensive and readily available. You just dip a strip into your prepared sanitizing solution and it tells you the concentration of available chlorine. This allows you to verify that your concentration is correct and safe, typically aiming for that 100-200 ppm range for no-rinse applications, ensuring you have enough to be effective, but not so much that you risk residues. It takes a guesswork out.
Test strips.
Yeah.
Okay, that seems like a smart, easy step for peace of mind.
Mhm.
Are there other sanitizers besides bleach?
Yes, there are others commonly used in the dairy industry. Efficacy. In terms of effectiveness, especially against biofilms, parasitic acid, often abbreviated as PAA, is frequently considered very effective.
Parasitic acid.
Yes. It's a strong oxidizer, and it's effective against a broad spectrum of microorganisms, including bacterial spores, viruses, and fungi, often more rapidly than chlorine. A key advantage is that PAA breaks down into harmless byproducts, basically acetic acid like vinegar, water, and oxygen. It also tends to interact less with any remaining organic matter compared to chlorine, meaning more of its sanitizing power is directed specifically at the microbes themselves rather than getting used up by tiny bits of leftover milk residue. It's often used in commercial settings and is available for farm use too, but again, following label instructions for concentration and use is critical.
Okay, PAA sounds interesting too. So we're cleaning diligently, we're rotating cleaners maybe monthly, and we're strategically sanitizing right before milking when needed, using the right amounts. We're putting in all this diligent effort. How do we actually know if all this hard work is paying off? How can we objectively verify that our process is truly effective? Because like we said, what you can't see, you can't always control, right? How do we prove our milk lines are actually kitchen clean on a microscopic level?
You've hit on probably the most crucial point for ongoing management: verification. Because you're absolutely right. The true measure of your cleaning protocol's effectiveness isn't just how shiny your stainless steel looks, but what the microbiology of your milk actually tells you. This brings us to a vital step for anyone serious about milk quality and safety. Verify your process with testing.
Testing the milk itself.
Exactly. Testing your milk provides direct feedback on the outcome of your entire process, from udder prep to cooling, including how well your equipment cleaning is working.
What kind of tests are we talking about?
Methods. The most common and effective methods for monitoring your cleaning effectiveness and overall milk hygiene are using coliform count, sometimes called a coliform plate count, or preliminary intubation count, PIC, and standard plate count, SPC tests.
Okay, SPC and coliform count, what do they tell us?
Let's break them down. Standard plate count, SPC. This test gives you a general count of the total number of viable aerobic bacteria present in your milk sample. It's a broad measure of overall bacterial contamination. A high SPC often indicates issues like poor equipment cleaning, inadequate milk cooling, or sometimes even underlying udder health issues like subclinical mastitis in the herd. For high-quality raw milk, a consistently low SPC is very desirable. Coliform count. This test is more specific. It measures the presence of coliform bacteria. Coliforms are a group of bacteria commonly found in the environment and in the feces of warm-blooded animals. Finding significant numbers of coliforms in milk is generally considered an indicator of contamination that occurred after the milk left the udder, usually pointing to issues with sanitation like improperly cleaned equipment, poor udder preparation before milking, or contamination during handling or cooling.
So SPC is overall bacteria. Coliforms point more towards sanitation problems.
That's a good general way to think about it, yes. Together, these tests provide quantifiable data, actual numbers on the bacterial load in your milk. This allows you to gauge the overall cleanliness of your entire milking process, including the effectiveness of your equipment cleaning protocols using real data, not just guesswork or visual inspection.
How often should we be doing these tests?
Frequency and benefits. Regarding frequency, a good starting point is to perform these tests perhaps at least once a month, just as a general checkup on your system. However, for truly active management, especially if you're troubleshooting a persistent quality issue, trying to really dial in your processes, or consistently aiming for the highest possible milk quality, maybe for selling raw milk where allowed, or for specific home uses like cheese making, doing them more frequently can be incredibly valuable, maybe weekly or even daily for a short period if you're making a specific change and want to see the immediate impact.
What are the benefits of testing that often?
Frequent testing allows you to identify trends over time, for example. Is your SPC count slowly creeping up month after month? That could indicate a biofilm slowly establishing somewhere. It helps you pinpoint potential trouble spots that might be developing before they become major problems. And crucially, it provides objective feedback, ensuring that any changes you implement, maybe switching cleaners, replacing old hoses, improving your drying method, have actually optimized your system for producing low-risk, high-quality raw milk. You can see the results of your efforts in the numbers.
That sounds really useful, but is it practical? Is testing expensive or hard to do for a small home dairy?
That's a great question because historically it might have been. But the good news is that on-farm or mail-in lab testing options for dairy producers, including small goat owners, have become increasingly convenient and much more affordable in recent years. There are various services where you can collect a sample according to their instructions, mail it in, and get results back electronically relatively quickly. Some local dairy labs or even vet clinics might also offer these tests. So this level of verification is definitely much more accessible than it once was. It really empowers you to see the microscopic, tangible results of all your diligent cleaning efforts, giving you valuable peace of mind and helping guide your practices based on actual data.
Wow, okay, that was a truly deep dive into the microscopic world, right inside our very own milkrooms. It honestly changes how you look at every single hose, every valve, every gasket, and every surface in there, doesn't it?
It really does shift your perspective.
Knowing what's actually happening at that tiny, unseen level, these bacterial cities building fortresses, makes you realize just how incredibly important every single step of our cleaning routine truly is, and how those small, consistent actions, day after day, can have massive ripple effects on everything.
Absolutely, and that's really the core message we wanted to convey today. The key takeaway here, strongly reinforced by the latest research like that Chilean study, is that hoses and equipment, especially those often overlooked components used for diverted milk, colostrum, or transition milk, are significant, frequently neglected sources of biofilms and potentially pathogenic bacteria.
So don't ignore the dump bucket lines.
Please don't. Therefore, thorough, consistent, and well-designed cleaning and sanitation protocols for all surfaces that come into contact with any milk or colostrum, not just the milk destined for the main tank or human consumption, are absolutely crucial. Overlooking these seemingly minor areas can genuinely have profound, long-lasting negative impacts on your herd's health, the quality and safety of your milk, and can even contribute to those broader public health challenges like the spread of antibiotic resistance.
So what does this all mean for you, our dedicated listener, tuning in today? Given how incredibly resilient and surprisingly impactful these microscopic communities are, and their potential to affect everything from the immediate health and well-being of your herd to the shelf life and taste of your delicious raw milk, and even the long-term effectiveness of life-saving antibiotics, we really encourage you to take a moment and examine your own daily routine with a fresh, critical eye.
Use this knowledge proactively.
Exactly. Ask yourself, what hidden corners in my milking setup might be harboring similar unseen challenges? Perhaps it's that one rubber hose that's looking a bit worn, or that valve you haven't taken apart in a while, or maybe that quick rinse step that really needs to become a full breakdown and scrub every single time. What small, consistent changes could you implement armed with the knowledge from this deep dive that could make a massive difference for your animals and your family? Take this information and use it to empower your own farm practices. Because really, when we understand the invisible world around us, we can take much better control of the visible outcomes we care about.
Well said. Knowledge is power in managing these microscopic communities.
We sincerely hope this deep dive on Hoofnotes, where we make goat science make sense, has helped you feel more informed, definitely more empowered, and hopefully more confident in your ability to keep your milk clean, your equipment sparkling, and your beloved goats healthy and thriving. Keep those questions coming our way, because the more we all understand, the better we can care for our amazing herds. Until next time, keep those hooves happy.
And those milk lines absolutely sparkling clean.